This week in lab, I have the pleasure of covering my favorite system, the nervous system. And specifically, we're going to talk about the central nervous system and microscopic and gross anatomy of the central nervous system. To begin, when we look at the brain in a frontal section here, what we see is some light areas and some darker areas. The light areas are called white matter, while the dark areas are called gray matter. Now, what gives these areas of the brain their distinct look is the presence of myelin. So myelin is actually a coating around the nerve axons that is white in appearance. So when we look over here and we see this lighter area, this is an area of the brain that has myelinated axons, while out in this area, we don't have myelin, and for the most part, we have neuron cell bodies. These are both considered areas of neural tissue. Now, in these areas of neural tissue, we have cells that are non-neuronal. So these are called neuroglia or nerve glue. And they're cells that occupy the space between neurons. It's been estimated that there are roughly 36 billion glial cells, which comprise 80 to 90% of the cells in the CNS. Neuroglia differ from neurons in several ways. They do not form synapses, have essentially only one type of process, retain the ability to divide, and are less electrically excitable than neurons. Now, neuroglia are divided into two major categories based on size: the macroglia and the microglia. The macroglia consist of astrocytes, oligodendrites, and ependymal cells, while microglia are their own type of cell. Astrocytes function to support neurons in their immediate vicinity. They provide a physical barrier between cells, maintain the ionic and pH equilibrium of the extracellular space around the neurons, and continually modify the chemical environment of neighboring cells. Oligodendrites are responsible for producing the myelin that makes up the myelin sheath around the axons. Now, in the peripheral nervous system, we have cells called Schwann cells that are responsible for producing myelin out in the periphery. Then ependymal cells actually line areas of space in the central nervous system, so the ventricles of the brain or the central canal in the spinal cord. And these ependymal cells actually are in contact with vasculature, and they're responsible for producing and circulating cerebrospinal fluid, which is the nourishing and protective fluid that we find around the brain and around the spinal cord. A couple of great slides that I found online actually show these glial cells. And here we can see astrocytes actually making a connection with a blood vessel and then in close proximity to a neuron. So here we see a neuron cell body. Then in this slide, we actually see another neuron cell body, or a soma, and an axon. And here we can see the oligodendrite, which is forming the myelin sheath around the axon. Can you guess whether we're in the central nervous system or the peripheral nervous system? That's right. Because it's an oligodendrite, we know that we're in the central nervous system. Now here's an artist's drawing of a neuron. And this neuron has projections that are coming into the cell body. I say coming in because this is actually the direction of flow of stimulus. So here, if we had some type of stimulus, we actually have the direction going towards the cell body, which would then come into a narrowing area called the axon hillock, and then get down to the axon and proceed down to the other end where we have synaptic end bulbs that can make connections with other tissues or other neurons. So underneath a microscope, we can see a nerve axon that is coated with myelin, and we have a break in this area called a node of Ranvier. Now here we can see Schwann cells in a slide, and what we see here is the nucleus of a Schwann cell, and then the myelin sheath surrounding an axon. And here's a closer look, and here we can see the numerous layers of wrapping around an axon. Now, in this slide, we're actually looking at the organization of a nerve bundle. So when we see a nerve bundle in a cadaver or in a picture, what we're actually looking at is we're looking at axons wrapped in a myelin sheath, then covered by a covering called endoneuronium, and this is an area that we call a fascicle. So right here is the entire fascicle, and then this fascicle is going to be wrapped in an area called perineuronium. Finally, we have bundles of fascicles that also have blood vessels running within them, and this entire area is called the epineuronium. In this slide, we're actually looking at a nerve bundle, and this is a group of myelinated axons. The white covering on the outside is the myelin, and the axons are in the middle. The analogy of a line of conduit is a good one, because here we have wires inside insulated coverings that we want the signal to stay within this wire. So this insulation prevents the signal from either dropping off or going into one of the other wires. So myelin serves basically the same purpose within our body. Now here we can see that there is an axon that is unmyelinated. And over here we can see a myelinated axon with many layers of myelin on the outside. In this slide, we're looking at a group of neuron cell bodies, and we can actually see the dendrites, or the processes, coming off of these neuron cell bodies. These dendrites would actually make connections in certain circumstances with other axons. So here what we see is dendrites coming into a neuron cell body, the axon coming down to what we call synaptic end bulbs, and here we have other dendrites that would receive a signal from these synaptic end bulbs, and then this would pass on to the next cell body and then down the next axon. And if you ever take a neurophysiology class, you'll use this simple representation diagram to draw neural networks. Now here we can see the spinal cord cut in a transverse section, and in this section what we can see is that the organization of white and gray matter is different from that in the brain. So what I'm saying here is that in the brain, the gray matter is on the outer regions, while the white matter is in the inner regions. And in the spinal cord, we find the opposite of that. So here we can see the gray matter in the center and the white matter on the outside. Just a couple structures to look at here. We have a spinal nerve, and this would bring us out to the peripheral nervous system. Then we have a posterior root ganglion, and this is where we find neuron cell bodies of the afferent neurons. And here we have a posterior root and then posterior rootlets. On the other side, we have anterior rootlets and an anterior root, and then the spinal nerve. And here what we see is that outside of the spinal cord, we have a number of layers covering the spinal cord. So here we can see the first layer is a delicate layer called the pia mater, right up against the neural tissue. Then outside of that, we have a spider web type of covering called arachnoid mater. And then finally, we have a durable covering on the outside called dura mater. in this gray matter area in the center, we have dorsal horns and ventral horns. Now, just a little bit about functional organization of the nervous system. So we have the sensory or afferent division, and this is transmitting information from the periphery to the CNS. This can be further broken down to a somatic sensory system and a visceral sensory system. The somatic sensory system is going to be information received from our skin, our fascia, our joints, skeletal muscles, and special senses, while our visceral sensory system is receiving information from our viscera, or what's in the area of our stomach, our blood vessels, those types of places. Things that we're not always conscious of what's going on, or most of the time not conscious of what's going on. Over on the other side, we have the motor or efferent division. And the motor or efferent division transmits information from the CNS to the rest of the body. Now, this can be further broken into the somatic sensory system, or somatic motor system, and the autonomic motor system. Now, somatic is going to be the voluntary movements that we think about when we want to move a muscle and we create an action. The involuntary system is going to be innervating things like cardiac muscle, smooth muscle, and glands. Now, knowing that, we can look at the spinal cord again, and what we can see is that we have afferent nerves coming in through the posterior root where we find the cell bodies, and then into the dorsal gray horn, and then out on this side, the ventral gray horn, to these efferent nerves going out to the periphery. Now, just to break this down a bit more, we can see that this is making connections with other neurons in this gray matter. So as I mentioned before, the neuron cell bodies are found in the gray area, while the myelinated axons are found in the white area. And this breakdown over here is showing you where you would find the different cell bodies for the somatic, visceral, autonomic, and somatic systems, broken into the sensory nuclei on the backside and the motor nuclei on the anterior side. In this diagram, we're just looking at the gross anatomy of the brain, and here we can see that there are two hemispheres, the right cerebral hemisphere and the left cerebral hemisphere. Now, this is color-coded to show you the different areas or the different lobes of the cerebrum. Your sheep brains will not be color-coded. Here what we're looking at is the frontal lobe in yellow, the parietal lobe in red, and the occipital lobe in blue. On these pictures on the side here, we can see the frontal lobe in red and the parietal lobe in blue, occipital lobe in green in the back here, and then this lobe is the temporal lobe on the side. A couple of other important names we have here. The things that kind of look like noodle-shaped pieces of nervous tissue are the gyrus, or gyri plural. And the grooves in between are the sulci, or sulcus, plural. Right down the center we find a division that actually splits the right and left cerebral hemispheres in half, and this is called the longitudinal fissure. Then we also have a sulcus that divides the frontal lobes from the parietal lobes, and this is called the central sulcus. Just looking at some other external structures of the brain, we can see up here this is the cerebrum, and then down below we have another area that's called the cerebellum, while the brainstem is down in this area, and the brainstem can be further divided into the pons, medulla oblongata, and the spinal cord. And here's a look back at a cross-section of the brain in the frontal plane, And here we can see the white matter in the center area and the gray matter on the outside. Now, a useful but silly analogy is the idea of the white matter being like highways, where you're trying to get someplace fast. And the gray matter, maybe that's like a coffee shop, where you might go there to interface or network with someone else. So the areas of gray matter, we can make connections with other neurons, while the areas of white matter, we just have fast-moving signals from one place to another. This week, when you come into lab, you'll be doing your first dissection of the year, and we'll be dissecting a sheep brain and beef spinal cord. I just want to go through the basic equipment that we'll be using and how we'll handle different situations in lab related to dissection. So the first thing I'm going to do is choose a size of gloves that fits me. We have small, medium, and large gloves, and we want the gloves to go onto our hands and fit comfortably. So that we can basically get our fingertips all the way into the ends of the gloves. So I've set up my gloves like this. We'll always wear gloves when dissecting. The specimens, in most cases, will be preserved specimens, and those contain harsh chemicals that will actually can potentially do damage to your skin. So you're going to want to wear gloves all the time while dissecting. I just want to briefly walk through where you're going to find different supplies in the lab here. So your instructor will have laid out basically a tray with dissecting supplies. We have a number of different things here that we'll be using. First, we have a couple different types of scissors. We have a scissors that has a sharp tip, and we have a scissors that has a blunt tip. Now, the blunt tip scissors is best used if we're trying to preserve part of what we're cutting. So we may want to, this week, preserve the meninges as we cut into the beef spinal cord. We'll be cutting off the dura mater, or the outside layer, and using this blunt end on the inside of the spinal cord will help us to keep the specimen in better shape. When we're just trying to cut off certain areas of a specimen, remove fat or remove some other type of tissue that we're not interested in having there, the sharp scissors will work great for that. Now, we also have a couple different types of tweezers. So we have a tweezers that actually has a claw type of tip, and this claw type of tip is good for pulling apart different things. So with the beef spinal cord, we will actually be able to lift up the different layers of meninges with that tip. And we have a sharp tip tweezers as well, and this will enable us to reach into small areas and remove things or pull things back. We also have two different types of a blunt probe and a sharp probe, and the blunt probe is going to allow us to not only point to something without destroying it at all, but it will enable us to get within a layer and to delicately move it back. Commonly we'll use a sharp probe when we're actually trying to pull back a layer or poke a hole through a certain area. So the sharp probe can be used, but be aware that this can actually dig into your specimen or whatever you're trying to dissect. And finally, we have scalpels, and the scalpels are of course very sharp. They attach on here with the blade that slides on, and there's quite a process to getting this on there. So if you do tend to break a scalpel in any way, you want to talk to your instructor about replacing the blade. These can be tricky to deal with, and as I said, they're very sharp, so when they do cut, they cut very deep. So you want to be cautious of that. If you do have a scalpel blade that's laying around, we do want to get rid of this in our sharps container here. So we'll just drop this in the sharps container, and that way no one is at risk of getting injured with that. And your instructor can replace the blades for you. So there's different scalpel blades for different size scalpels that they'll put on there depending on what size scalpel you have. At each of the different stations, you've noticed that you have skeleton labeled for the drawer with all your bones, and generally right above that we're going to have a tray drawer. So this tray is actually a large dissection tray. And this tray will enable you to have enough space to work with all the different things that we'll be dissecting this year. So this tray is easily washed. When you're finished using the tray, we want it to be just as clean as it was when you found it for the next set of people in lab, so please do clean it up. What we will do is we'll bring these over to the sink and we'll actually use some of our dish soap and a green scrubby, basically, which will be in one of the drawers near the sink. The green scrubbies will probably be cut in half because you only need a small portion to do this cleaning. So please use this to clean up, and we want our sinks to stay clean as well, so when you're done with any of the dissecting supplies that may get in the sink, please just take a paper towel and wipe out inside the sink, and then clean that, or bring that over to the trash and throw it out. All of the dissecting supplies that are preserved can go right into the trash. We do take care of the inorganic liquids and use proper disposal methods for those. But all of the bodies, the brains, spinal cord can be just put right into the trash. And of course, once you've washed your hands, or once you've taken off your gloves, which there's a very specific technique for this, we are going to dig our thumb right into the palm side of your glove, and you're going to remove the one glove and then do the same thing on the other side. And that way we are preventing any type of contamination getting on our hands. And of course we'll throw that away and we'll come over and wash our hands. This is medical grade disinfectant soap, so we will use this and clean up our hands. And dry them off. And of course, if you want to double down with hand sanitizer, that's okay with us. So you have hand sanitizer right over there. Now, should you get anything from the dissection supplies, any of the liquid into your eyes, we do have a hand wash, or an eye wash right here, and the eye wash will basically allow you to rinse all of that solution away from your eyes. In the case of a serious event where we had a bigger spill than that, we do have a drenched shower too. And this is not something you want to pull accidentally, because an awful lot of water will come down on your head and it'll get all over the floor as well. But in an emergency, that's what this is here for. Most of the chemicals and things that we work with, there's not much of a risk for that, but it is a precautionary method that I do want you to be aware of. So this week, again, we are going to be dissecting the brain from a sheep and the spinal cord from a cow. And we have plenty of these supplies, so as many people want to dissect can. So please enjoy the dissection next week. And as I mentioned, there will be a whole lot more next semester that we'll actually be doing as well. See you next week in lab.
Here's the sheep brain with the meninges still attached. The meninges consist of three membranous layers that surround the brain and spinal cord. The inner layer is the pia mater, the middle layer is the arachnoid, and the thick white encasement seen here is the outermost layer, called the dura mater. To remove the meninges, I like to cut around these ventral structures, because otherwise they're easily pulled off with the meninges. Taking a look at the dorsal surface of the sheep brain, we can see the cerebrum, The cerebrum is covered with convolutions, elevations, called gyri. Separated by shallow depressions called sulci. The cerebrum is divided in half by the longitudinal fissure. Into the right cerebral hemisphere and the left cerebral hemisphere. The cerebral hemispheres make up over 80% of the brain. The activities of the right and left cerebral hemispheres are specialized for different functions, making them unique from each other. The cerebrum is separated from the cerebellum by the transverse fissure. As I mentioned, this is the cerebellum. The cerebellum is involved in motor coordination and balance. In this video, I'd like to examine the brainstem in a few different views. Let's start with the ventral surface, where we can see all three parts of the brainstem. This is the midbrain. The pons. And the medulla oblongata, also known as the medulla. Let's look into the transverse fissure, which is in between the cerebrum and the cerebellum. These four bumps make up the dorsal surface of the midbrain. This area is called the corpora quadrigemina. The corpora quadrigemina is made up of the two superior colliculi, which are visual reflex centers, and the two inferior colliculi that relay auditory information to the sensory cortex and are involved in reflexive responses to sound. Lastly, let's take a look at the brainstem in a mid-sagittal section. Here is the transverse fissure, and just anterior to the transverse fissure is the superior portion of the brainstem, a region called the midbrain. We can see a superior colliculus, here's the cerebral aqueduct, Let's take a look at the diencephalon in a mid-sagittal view. The diencephalon makes up the central core of the brain. It's made up of three regions. The thalamus, which is a central region, filters information ascending to the cerebral cortex. The hypothalamus, which monitors various conditions in the body, plays an important role in homeostasis. The hypothalamus contains the mammillary body. To make a frontal section, what we want to do is make an incision to divide the brain into an anterior half and a posterior half. Taking a look at the frontal section, we can see the gray matter that makes up the cerebral cortex. Gray matter consists mainly of neuron cell bodies. Right underneath, we can see the white matter, which is predominantly myelinated axons. Here's the longitudinal fissure, which divides the right and left cerebral hemispheres. This is the corpus callosum, connecting the right cerebral hemisphere to the left. And right underneath that are the lateral ventricles, which are separated by the membrane, the septum pellucidum. To make a mid-sagittal section, what we want to do is cut right down the longitudinal fissure and divide the brain on midline into two separate halves. Let's take a look at the mid-sagittal section of the sheep brain. We can see the cerebrum, and this white structure is called the corpus callosum. The corpus callosum is a commissure. It consists of myelinated axons that connect the right and left cerebral hemispheres. Right below the corpus callosum is a lateral ventricle. There's a lateral ventricle on the right and on the left. They're lined with ependymal cells that circulate the cerebrospinal fluid found inside of them. This central region of the brain is called the thalamus. Right below the thalamus, this region is called the hypothalamus. This is the optic chiasm, where some of the right and left optic nerve fibers cross midline. This round structure is the mammillary body, which is involved in the transmission of olfactory information. Here is the pituitary gland. This is the pineal body, which secretes one hormone, melatonin. Here we can see the transverse fissure that separates the cerebrum from the cerebellum. And moving through the transverse fissure, we look right onto the dorsal surface of the midbrain. This projection is called the superior colliculus. It's part of the corpora quadrigemina. Taking a look at the anterior portion of the midbrain, let's take a look at the ventral surface of the sheep brain. This is the inferior surface of the sheep brain. These are the olfactory bulbs, the optic nerves, the optic nerves originate on the back of the eyeballs and carry visual information into the brain. Some of the optic nerve fibers cross midline at the optic chiasm and then continue on into the brain through the optic tracts. This is the pituitary gland. It consists of two lobes that secrete hormones. Some of those hormones have direct effects in the body, and some serve to regulate other endocrine organs. The pituitary gland is attached to the hypothalamus by a stalk called the infundibulum. Posterior to the pituitary is this white elevated region. That's the mammillary body. Moving on into the brainstem, this is the anterior. Let's take a look at the ventricles inside of the sheep brain. The ventricles are cavities within the brain that are interconnected. They're lined with ciliated ependymal cells, which circulate cerebrospinal fluid. Right underneath the corpus callosum, we find the lateral ventricle. There are two lateral ventricles, one on the right and one on the left. The lateral ventricles are separated at midline by a membrane called the septum pellucidum. This region is the third ventricle. You'll notice some pink feathery tissue. This is called the choroid plexus. The choroid plexus is a vascular tissue covered with ependymal cells that produces cerebrospinal fluid. The choroid plexus is found in all of the ventricles. The fourth ventricle is just anterior to the cerebellum. The medulla and pons form the